Hello students, this is Kanika. Welcome to my channel PrepBiology. So today in this class, we are going to discuss the law of independent assortment under the Mendel's laws of inheritance. In previous two classes, we have discussed law of dominance and law of segregation. In today's class, we are going to discuss law of independent assortment. So as we all know, law of independent assortment comes under the inheritance of two genes. Okay, so what is the meaning of inheritance of two genes? The study of the inheritance of two character that is the two pairs of alleles that is called the inheritance of two genes. Okay, so under this we have the law of independent assortment. What it states? The law states that when two pairs of traits are combined in a hybrid, why two pairs? Because we are talking about the two characters. Okay, characters. So when the two pairs of traits are combined in a hybrid, segregation of one pair of character is independent of the other pair of characters. So when the gametes are formed, when they segregate or separate from the other pair of character, their expression is independent from the other characters. Okay, so this is the law of independent assortment. This law of independent assortment is explained by the dihybrid cross. Di means where the two characters are considered in a crossbreeding. That's why it is expressed by the dihybrid cross. So in dihybrid cross, what happens? We have cross the two, we will take the two characters. Okay, so understand it carefully. We take the two characters of a plant, that is the seed color and seed shape. Okay, so we know that according to the Mendel, under the seed color we have two traits, yellow seed color and green seed color. Yellow seed color is dominant and green seed color is recessive. Yellow is dominant, that's why we write it as capital Y, and recessive, that is small y. Second character we take is the seed shape. According to the Mendel, the dominant character of the seed shape is round, that is capital R, and the recessive character of the seed shape is the wrinkled, that is small r. Okay, so these two characters Mendel take and he cross pollinate. Okay, so when we cross the one parent with dominant character, that is round seed and yellow color, because these are dominant, so capital R, capital R, capital Y, capital Y. So when we take this other parent which is fully recessive, that is wrinkled and green. Wrinkled small r, green small y, small y. When we gametes are formed, they have capital R, capital Y, and they have small r, small y. Fusion takes place, they form capital R, capital Y, small y. I hope this is clear to you all. So this is the F1 first filial generation. And what are the kinds of seeds we see in F1 generation? Capital R is dominant over small r, so capital R represent the round. So round, and capital Y dominates over small y, capital I shows the yellow seeds. So this is the F1 generation. Now we take the selfing. Selfing takes place and forms, it will segregate their gametes, capital R, capital Y, small y. Now fusion takes place, capital R, capital R, capital Y, capital R, capital Y, capital R, small y, capital R, small y. Now small r, capital Y, small r, capital Y, small r, small y, small r, small y. So we have after the segregation, they forms this. Now, in dihybrid cross, we write these in different, like capital R, capital Y, capital R, capital Y, capital R, small r, capital Y, small r, capital Y, small r, small y, small r, small y. These are the gametes of one parent. Now, other parent is, suppose this is the male and here is the female. So, capital R, small y, capital R, small y, capital R, small y, small y, small r, capital Y. So, we write these gametes like this. Now, the fusion takes place. Okay, now fusion takes place. So, capital R, small y, capital R, small y form the capital R, capital R, capital Y, capital Y. Capital R, small y, capital R, capital Y. So, capital R, capital R, capital Y, small y. Capital Y, this is capital Y and this is small y. Now, this with this. Capital R, small r, capital Y, capital Y, capital Y, capital Y. So, like this, the fusion takes place. Okay. So, capital R, capital R, capital Y, small y, capital R, small y, small y. Now, capital R, small y, small r, capital Y, small y. Now, capital R, small y, small y. Next, capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y. Now, this with this. Small r, capital Y, capital Y. Small r, capital Y, small y. Capital R, small y, capital Y. Capital R, small y, small y. Small r, capital Y, small y. And small r, y and small y. So, these are the offsprings we get. These are the phenotypes of the offsprings we get the F2 generation. So, this is the F2 generation. Okay. So, if you see now the ratios we found, now understand it carefully. We have phenotypic ratios. Okay. Phenotypic ratio. So, first, we found that full dominant. Okay, so we have the full dominant is capital Y and small y, capital Y and capital R. This is the full dominance. So we have right here round, that is capital R, and yellow, that is capital Y. Next, we write the full recessive, that is wrinkled, that is small r, and green, that is small y. Okay, now we have mixture of characters. We have a round, that is capital R, and green, that is small y, and yellow and wrinkled. Yellow, wrinkled, that is small r, and yellow is dominant, capital Y. Okay, now first count the round and wrinkled. Okay, first we found the round and wrinkled, capital R and capital Y. Okay, capital R and capital Y. Here capital R, capital Y 1. Here capital R, capital Y 2. Capital R, capital Y, yes, 3. Capital R, capital Y 4. Capital R, capital Y 5. Capital R and small y, no, we do not count here. So capital R, capital Y 6. Capital R, capital Y is here, no. Capital R, capital Y 7. Capital R, capital Y 8. Small r, capital Y, no. Small r, capital Y, no. Capital R, capital Y 9. We have 9 round and yellow. seeds. Now let's discuss the pure dwarf, that is small r small y. Let's count it. Now these shaded, that 9 seeds that we shade, we do not count in next counting. Okay, so the left one we will count. Okay, so we require small r small y. Here small r small y, no. Small r small y here is there, but capital R dominates over small r. Here capital R capital Y, no. Small r small y here are present, but capital Y dominates over small y. So we have a pure dwarf is small r small y only one. So pure dwarf is small r small y is only one. Okay, now we count for round and green, capital R and small y. Capital R and small y. This is capital R and small y 1, capital R and small y 2. Here capital R, no, no. Capital R small y 3. So we have 3 round and green. Now the left of 3, these are the wrinkled and yellow. Wrinkled small r, yellow for we required the capital Y. Small r capital Y, yes. Small r capital Y, yes. Small r capital Y, yes. So like this we complete the ratios. So we have 9 ratio 3 ratio 3 ratio 1. Okay, 9 ratio 3 ratio 3 ratio 1. So this is the phenotypic ratio of dihybrid cross. Like this dihybrid cross we do. Okay, so practice, take different characters and practice by those characters, practice this dihybrid cross. The method of doing dihybrid cross is this only. Okay, by this easy method you can do the dihybrid cross. Okay, so the ratios, the phenotypic ratio is a 9 ratio 3 ratio 3 ratio 1. Okay, 9 is a full dominant and 1 are full recessive. So by this the law of independent assortment is explained. Okay, so dihybrid cross is very important. So make sure practice the different crosses. Okay, so if this lecture is beneficial to you all, like, share and subscribe my channel, that is PrepBiology. Keep studying, keep learning. Thank you so much.